I'll start introducing the first speaker, who's going to be from one of these companies building cakes from all of these ingredients, and that's ISABEL AZNAREZ from Stoke Therapeutics.
Can you hear me okay? Yes. Thanks so much, Anna, for the introduction, and thanks to the organizers, to Mike, for inviting us here to present our technology. Can you guys see me from above this? I need a little stool. No, I'm joking. I'll step back a little. So I'm going to tell you one of those ingredients and tell you about our TANGO technology. Let me see if I can advance. It did. I have to make disclosures, and I'm of course an employee of Stoke, and I'm going to make some forward-looking statements as part of this presentation. So with our TANGO technology, we aim to address the underlying cause of genetic diseases. And like Anna said, that is the first thing you need to know to be able to develop therapeutic approaches. What is the gene that causes disease? Syngap, we know. And so we develop these therapeutic treatments by targeting RNA. And I'll tell you a little bit more about that in a few slides. So with our treatment, with our method, we aim to increase functional protein from the functional copy of the target gene. In the context of haploinsufficiencies, there's a functional copy there that is a great, reservoir of good protein for us to leverage and increase gene expression. By targeting the functional copy of the gene, we're able to address a disease that is maybe caused by many different mutations, like Syngap is, but because we're leveraging that wild-type copy, we're able to develop one treatment for the entire patient population, provided that the mutant copy is a loss-of-function mutation. This TANGO technology is also inherently cell type and tissue specific, and we heard today about how it is important to know the function and to know where Syngap is expressed. And here, because we're acting at the level of RNA, we're only upregulating Syngap or any other potential target protein in the cells and tissues where that gene is expressed. There are no limitations in the size of the gene. Syngap gene, SCN1A genes, they're very large genes, and there are certain technologies that may not be able to address these diseases, but in the context of our technology, we don't have to worry about that. So before I go into the details of how this works, I want to step back, way back, and go through the central dogma in biology, which is the DNA, which forms all our genes, the genes in our body, which they get copied into a messenger that will take that message, that code on how to make all the proteins in our body to the protein factories. And so the message, or the RNA, is what our technology focuses on and targets. And so the RNA, after it gets copied from the genes, it gets heavily edited by a process called pre-mRNA splicing to form that actual messenger RNA that carries the code to make proteins. And this is the step where we focus on, the editing. And that editing, which we call pre-mRNA splicing, ensures that the coding regions in a gene, which we depict by rectangles, get joined together, and the non-coding regions, which are the lines or the introns, are removed from the pre-mRNA to make that mRNA. But this process can happen in many different ways. It's called alternative splicing. And so in this particular example, we have this pre-mRNA that can be spliced in different ways. One way is to generate a bandana mRNA that will code for a bandana protein, but it can also be spliced in a way to skip that D exon and code for an mRNA that will encode a banana protein. And this is what we call productive splicing. From a pre-mRNA that gets spliced, a protein is going to be made. But there's something called non-productive splicing, or at least that's how we termed it, which is when non-coding exons are included in the final mRNA. And when this happens, it breaks the mRNA, it breaks the meaning, introduces a premature termination signal, and that RNA is non-productive, will not generate any protein. This occurs naturally in the cells. This is not a disease-causing event, and often regulates expression of genes. So when that gray exon or non-coding exon is included, that transcript is going to be degraded, and again, no protein is going to produce. So understanding this mechanism is important to understand how our TANGO ASOs work. So in the context of haploinsufficiencies, again, like SCN1A or Syngap, you have one normal copy and a mutated copy, and the disease is caused by a reduction down to 50% of protein levels. We all know that. So in this particular example, we have a pre-mRNA that contains one of those non-productive exons, that blue NMD exon, sometimes called also poison exons. And this pre-mRNA it can be spliced in a productive mRNA and a non-productive mRNA, and the non-productive mRNA will be degraded. And that, again, not related to the disease, it happens naturally. So with our TANGO ASOs, we're able to target that non-productive exon and prevent its inclusion in the final mRNA. And by preventing this inclusion, we almost exclusively generate productive mRNA from both alleles, the wild type and the mutant. Now, the mutant allele still won't do anything productive, but the increased levels of productive mRNA from the wild type allele, from the normal allele, will lead to more protein production, and that increase in protein production makes up or compensates for the loss of the mutant allele. And that's the sort of the TANGO concept. That's what we call the TANGO concept. So using that concept, we found many different genes that have those non-productive exons that we can target and leverage to increase gene expression. And these are the targets that we're currently working on. We're working on SCN1A for Dravet syndrome, and that's our most advanced target, and the one that I'm going to talk to you about today, because I can go from the very beginning of preclinical data all the way through a summary of our current clinical studies. As you all know, we're also working on developing a treatment for Syngap, but it's very early stage, and we want to be able to tell you the story once we have a complete story to tell, basically. So in the context of Dravet, as many of you know, are very aware about Dravet syndrome, it is a haploinsufficiency, and it affects 1 in about 16,000 babies. And we saw earlier today from Ingrid that there's a high incidence of SUDEP in patients with Dravet syndrome. About 20% of kids with Dravet syndrome will not reach adulthood. And the vast majority of patients suffer from seizures, very severe seizures, status epilepticus. But unfortunately, the majority of these seizures, the majority of the patients don't have their seizures controlled. 90% of patients don't have their seizures controlled. Dravet is also more than just seizures, and there are other comorbidities that affect these patients and their families, and these are quotes that we received from families from Dravet patients. And clearly there is an intellectual disability component, language and speech disturbances, sleep abnormalities, and movement imbalance disorder. And we aim, by targeting the root cause of the disease, to address not just the seizures, but the entire disease. So we first tested our antisense oligonucleotide, which is our modality, in a Dravet syndrome mouse model. And in this model, we injected a single dose of STK001, which is our clinical candidate, at postnatal day 2 in these mice. And then 7 weeks later, or 14 weeks later, we looked at the protein level. That's the first thing we looked. Are we able to restore protein levels? In this mouse model, we can also measure SUDEP, we can measure survival, and we can measure spontaneous seizures. So let me show you first what happens at the protein level. So the protein level in dark gray or dark blue, I'm not sure how it shows there, it's the untreated or placebo-treated mice. In both 7 weeks and 14 weeks, we see approximately 50% of protein levels, which is what you would expect from a haploinsufficiency. Upon treatment with STK001, a single dose, we can see that at 7 weeks and 14 weeks, we are able to restore the levels almost back to normal, almost back to 100%. That's what the line is showing, by targeting that non-productive exon in SCN1A. So what does this restoration of protein levels does to the survival phenotype of these mice? As you can see in the red line correspond to the placebo or PBS treated mice, whereas the orange line correspond to the STK001 ASO treated mice. As you can see, all but one survived through the observational period of 90 days. So again, what the restoration of NAT1.1, which is the protein that is encoded by SCN1A, leads to rescue of the survival phenotype in these mice. We also looked at spontaneous seizures. And we implanted electrodes into the brain, and we measure spontaneous seizures at 2 different time points. A time point where the disease is starting in terms of the phenotype, the seizure phenotype is starting, and then a later time point where the mice, some of them have died, of course, and we can't measure, but then those that survived, we measure and see how are the seizures being controlled. And as you can see here in both graphs, at both time points, we see a significant reduction in seizure frequency in mice. The gray is untreated or placebo-treated, the yellow or orange is the ASO treated. We see a significant reduction of seizure frequency. So with a single dose, a single dose of STK001, we are rescuing the survival, and we're also reducing significantly the spontaneous seizures in this mouse model. So to summarize the preclinical package that supported the clinical development of STK001, in a Dravet mouse model, we're able to restore protein levels back to normal for more than 3 months. The restoration of protein levels led to a significant reduction in mortality in this mouse model and a significant reduction in seizure frequency. I haven't showed you in the interest of time, and I have no idea how I'm doing with time. And I cut a lot of slides. I could have added. I could have kept them. Okay. I haven't shown you this data, but in non-human primates, when we tested this STK001 ASO, we saw a very broad distribution in the brain, and we administered it in the same way that we're treating patients right now, which is via intrathecal administration by lumbar puncture. And in NHP models, in NHP, we're able to see also upregulation of NAT1.1. And of course, NHPs are wild type, they don't have any disease, but we're able to express above levels of normal NAT1.1, and it seems to be a pretty safe. We have not seen any talks related to that. And then in NHP toxicology studies, we see that the STK001 is tolerated at the clinical doses that we're currently using in patients. So now, just a quick summary of what we're seeing in the clinical trials. We have 2 trials, one in the US and one in the UK. And now, in addition, the open label extension arms of both trials. And we have dosed patients between 10 milligrams and 7 milligrams, single dose and multiple doses, and they all seem to be generally well tolerated. Patients treated with 2 or 3 initial doses of 70 milligrams experience a significant reduction of 80% of seizure frequency at 3 months, and 89% at 6 months. There are more patients in this cohort that are going to come early next year. In the dose groups of 30 and 45 milligrams, we're seeing a maintenance of the reduction of seizure frequency, which it tells us that whatever effect that we're seeing initially is not a placebo effect. It's something, usually placebo peaks and then goes down. This is maintained with subsequent doses. And what makes me the most excited, I think everybody at Stoke, is that we're not only seeing an effect on seizures, but we're also seeing an improvement in the cognitive phenotype of these patients. And this is telling us that we're actually treating the entire disease and not just the seizure phenotype. And this is what makes me and everybody else at Stoke the most, and I'm sure the families, the most excited. We're measuring, and this is something that we had to establish ourselves. We're using 2 different scores to be able to measure cognitive development. And with those 2 scores, with both scores actually, we're seeing significant changes in cognitive measurements, and cognitive and behavior measurements. So to end the presentation, I would like to thank everybody in the Stoke team who contributed to these studies, our collaborators, Laurie Isom and Manoj Patel, who we work with to look at the survival phenotype and the spontaneous seizures. And of course, a special thanks to the patients and their families who have embarked in this journey with us and are brave to believe in us and do this with us and see what happens. All right, thank you.